have been using automation with a lot of our MAM workflows, and it does seem to be working pretty well most of the time. But sometimes even the automation technology doesn't seem to be quite there yet in terms of you'll get errors with your system. So we have had some challenges with that, but when it works and actually goes through, yes, the reproducibility is great. On that note too, though, there is still some other concerns, like from a compliance side, how do you, if there was a mistake in the prep or if something happened, how do you keep eyes on that? And I know there's been different, add-ons that some systems have, like a camera, so you can watch the prep all the way through. But then if you're doing that, you're not really time savings if something goes wrong, so it's a little bit tricky to implement with those other considerations, but definitely add automation to the future. So it's something that we're definitely keeping on the forefront with our things here. So any of the assays we develop, we try to keep automation in mind to make sure it's compatible.
Yeah, I mean, I think automation is in sample preparation, frankly, is one of the key steps to any mass spec experiment, as we probably all know. And automation is going to play a key role in MAM implementation and manufacturing and quality. I mean, I think it also needs to be thought about in terms of integration and automation, because we're going to want to integrate in time directly into, for example, into single-use bioreactors and have those technologies be automated. It's interesting, though, because we do a lot of work in comparing automated sample preparation and technician sample preparation. I think the automated sample preparation in many cases for us is clearly more reproducible. But in some instances, depending on which process we're using in the automated sample prep, we see significantly more miscleavages with the some of the trypsin assays. So it's really interesting, right? To ensure that you weigh those sorts of effects on the molecule against the reproducibility, and as far as I'm concerned, as long as the method is reproducible almost 100% of the time, then I can live with some miscleavages compared to what we might be able to do in solution.
I agree with both Rachel and Jared in the sense that automation is the key for sample preparation. For MAM to be applicable to in QC, manufacturing, at all the places, at all the stages of the product life cycle, it's really important that harmonization is the key. So sample preparation is an important aspect. Even for USP is working on a draft chapter, working on the best practices of MAM. So some initial work has been done to optimize the processes, like, what are the different conditions for the sample preparation. Having said that, the automation would help in the long term, in the sense that the real need of the MAM is to have high throughput analysis. So all these conditions of sample preparation, the different, even as small as how much trypsin is needed, what kind of trypsin is needed, what pH, what temperature. So all those of small different steps of digestion procedure, if that needs to be optimized, that needs to be harmonized, would say. So that's where, having automation would help in optimization of this and minimizing the variation when you are monitoring it across the labs or across the sites of the same manufacturing. So that's where I think for having a high throughput MAM applicable to multiple sites, it's really necessary to have the automation in there.
Sample preparation is a real challenge today. Even if you have the best LC-MS systems that are running perfectly, if your sample prep is not robust, you won't have good results. So for MAM, for example, the deamidation, oxidation can be induced during sample preparation, and depending on how you do your sample prep, especially if you do it manually, these artifacts can be quite annoying. So you really need to have a robust method. So the assessment of sample prep robustness is really critical if you want to use that in a QC environment or during stability testing in order to get reliable results all along the life of the product and of the method. Automation is clearly something that could be useful. In our case, so we work on many different projects, but not with a high throughput. So we do not work in process development. So using robots, very large robots, is not an option for us because it takes a lot of time to develop the method on a robot. So we'll probably go to more simple automation systems. And I think that's the key today to get reliable results and to push MAM into a QC environment.